All right, so we're going to move on to our next phylum then. And again, we're going to make a leap, quite a significant one, in complexity. New systems, more complex. So we're going to look at the annelids. That's phylum Annelida. So the annelids then have, firstly, let's go over their body plan, diagnostic characteristics. What about the symmetry? Bilateral symmetry. Layers. Embryonic germ layers. Triploblastic. In fact, everything else we talk about is triploblastic. That's as many of you can have. What about the cavity status? They are coelomates. So, bilateral symmetry, triploblastic coelomates. That's the annelids. The descriptive characteristics, they are segmented roundworms, or just segmented worms. So the body then is composed of segments, or sometimes you can think about them as a series of fused rings. And each segment is essentially the same as every other segment, but there is some specialization of the segments in the anterior segments of the animal. They've got a fairly well-developed closed circulatory system. Now in lab, we did an earthworm dissection, and you could very clearly see the blood, which was red, contained within vessels. So a closed circulatory system then, the blood is entirely enclosed within vessels. It doesn't come in direct contact with the tissues of the body. Okay? It's got a double ventral nerve cord. Now, if you were to open up an earthworm, you can tell the vessels very easily because the blood is red in an earthworm. You're looking for red liquid in vessels. What about nervous tissue? How can you typically tell nervous tissue? What colour is it usually? It's a whitish tannish colour. Yup, white tannish colour. Okay. So to figure out what are the nerve cords, you're going to look on the ventral surface, right? Ventral side. In fact, they're sort of run along the body wall. There's a double nerve cord, and they kind of look a bit like this. So here's one nerve cord. And here's the other nerve cord. And these are nerves that branch off. Okay? So you can always tell it's whitish in colour, and you can see it in the dissection. They have nerve ganglion. Now, in the dissection, you could very clearly see that there's this nerve ganglion right by the esophagus. And a nerve ganglion, think about this. If you've got a nerve, sometimes the nerves have a little thick or bulbous part. So anytime you see that sort of thickened or bulbous part to a nerve, it's called a ganglion. And it's usually where the cell body is, the nucleus part of the neuron. And that's why it's a bit bulbed out, because the nucleus takes more space. Okay? It's usually where the cell body or the nucleus is. So anytime you see that, it's a ganglion. They've got nerve ganglia. They've got a very well-developed gut, very well-developed digestive system. Definite mouth, definite anus, one-way passage through. And annelids are coelomates. Now they're hermaphrodites, which means what? In the same individual, you've got both male and female gonads. Okay? Ovaries and testes in the same individual. So the same individual can make eggs and sperm. But they tend to cross-fertilize. They don't fertilize themselves, they want to cross-fertilize another individual. What's the advantage of, you think, of being hermaphroditic? So if you fertilise yourself, you're right. You don't have to wait around for anybody else. But these tend to cross-fertilise.
More opportunity to reproduce.
Why is that?
Because they're more likely to run into something they can reproduce
Right.
than waiting for the right sex.
Every individual they run into, they can mate with. Just imagine if you're a rhino in Namibia, and there's hardly any black rhinos now. I mean, well, there are some, but there's not a lot. And your population density is super low. You wander around, you're a lone male, you're looking for a female, and maybe there's not one for miles, right? You've got to hope, in order to reproduce, that the next rhino you bump into is going to be a female. And it might not be. But in this case, you know you're going to mate with the next individual you bump into. What's a downside to being hermaphroditic? What's, a burden? What do you think?
Bearing young is a biologically expensive process.
So it is. In these cases, they release eggs, but you're on the right tracks. What could be biologically expensive about being hermaphroditic? Right, having both gonads, right? They're expensive to maintain. If you don't need them, they're expensive to maintain, right? There's a load, there's a cost to that. So there's costs and benefits of each. Okay, and they mostly live in damp and wet environments. Many live in the ocean, many live in fresh water, many live in damp soil. Those that live in the ocean and fresh water many of them tend to live in the sand or the mud or under rocks. Not a lot of them are free swimming, out with the fish, for example. Right. Why in this case is water and dampness important?
Absorption of nutrients?
Close. They've got a gut and they digest their food and they ingest food. But you're on the right tracks.
Gas exchange.
It's exactly what it is. They need to have a moist epidermis for gas exchange. Their gas exchange surface is their epidermis, and that must be kept moist. Gas exchange surfaces are always moist. Even the surface of your lungs are moist. All right. If they dry out, they literally suffocate. So I'm going to follow your book, the taxonomy of the annelids in your book to some degree, but I'm also going to deviate a little bit. So your book divides up the phylum Annelida into two groups, the Polychaetes and the Oligochaetes. Now here's what your book does. It groups the earthworms and leeches. So earthworms and their relatives and leeches, it groups those together into the Oligochaetes. Okay? And it leaves the Polychaetes separate. I'll, again, describe these classes in just a moment. You can break the Oligochaetes down and separate the earthworms from the leeches, and you can place the leeches in another class Hirudinea. So we'll go through class by class then. First class I want to go over are the Polychaetes. Poly means many. And these chaetae or setae are bristles or hairs. By the way, there's a bunch of different ways to spell chaetae. All right? I spell it just like that. Sometimes you can leave out the A. There's different correct ways to spell this, which is fortunate for you, right? Because you can get it wrong and maybe it's okay. So, members of the Polychaetes then not only have many bristles, many little hairs, I'll show you pictures of them. On each segment, they have these little paddle-like structures called Parapodia. Singular is Parapodium. And I'll show you pictures of them. Now that parapodium, that little paddle-like structure on every segment, increases the surface area of the segment. So that increases the surface area for gas exchange, and they kind of act a bit like gills. So the Parapodia serve two functions. One, increase the surface area for gas exchange. They kind of act like gills. The other function is they literally act like paddles. The animal uses them like paddles to swim for locomotion. Okay? And each parapodium has these little bristles made of chitin. Is that the same stuff that your hair is made of, or a little different? What's your hair made of? Keratin. What kind of a molecule is keratin? It's a good protein. Chitin is what? Carbohydrate, good. And it's found in the exoskeletons of things like insects and other arthropods, good. All right, and they've got muscles that are able to contract to move the chaetae in and out. How on earth can there be muscles that move them both in and out? Wouldn't you think a muscle contraction would just pull them in? It's kind of cool the way it works. If here's a parapodium, and here are some of those bristles that are sticking out, there could be muscles attached there to some other structure. And if these muscles contracted, that would pull them in, wouldn't it? Well, you can have other muscles attached there and there. If these muscles contract, then the bristles go out. You see how that works? Muscle, muscle, bristle. Contraction of this one pulls them in. Contraction of this one, they go out. Good? And obviously, if they're going to act as gas exchange organs, a bit like gills, they've got a rich supply of blood vessels, capillaries. Now, you'll have an opportunity to see some polychaetes in lab, all right? And you'll see the Parapodia very clearly. All right. Good? So many polychaetes then are marine, and you find many of them in the intertidal zone, and you find many of them in the sand or the mud or under rocks in the intertidal zone. Got any fishermen? Anybody likes to go fishing? I do. When I was a kid, I'd spent tons of time by the ocean, by the coast, and I would go out every tide out, and I would dig for worms, and I was primarily digging for polychaetes, and that's what I'd use for fishing bait. All right, so there's the intertidal. Get yourself a shovel or a fork, and you're going to find some annelids. Push over some of those rocks. Chances are you'll find some polychaete annelids. And there. Go to Rocky Point, and there's some areas of sandy beach that are, have a great rocky intertidal area, and there are tons of polychaete annelids there. One of them is quite notorious or infamous. It's called a fireworm. It even looks red, and those bristles, those setae, you pick it up, they poke into your skin, and they come out very easily. They're very irritating. It's a defense response. It's a bit like the glaucids on some of the cacti that we have. All right, so there's one of those polychaetes. See all the bristles? Not all of them. All of them have nasty, irritating bristles. They're usually quite nice, quite friendly little things, right? But you can see the Parapodia, the little extensions. I'll show you a clearer picture. And the bristles are on the end of the Parapodia. There, this one shows it really nicely. So all of these are clusters of bristles. Just in there is the Parapodium. We've all got one. Really clearly shown here. Oh, let me back up. These structures here, they just look like gills a little bit, don't they? They're Parapodia. So this is the head end of one of those polychaete worms. All these are little sensory structures. Okay. And there's another one. This one would stick these out, actually, and it would form a gas exchange surface. Okay. Now these tube worms, all right, that are often found at great depths, high temperatures and pressures near thermal vents are also Polychaete annelids. All right, I'm going to try one of these, and if it doesn't work, I'm going to give it up. Well, I'm giving it up. You can wrestle, find them. One of these should work. The YouTube one's usually good. So this is a ragworm feeding. Now you can see it's got a pharynx that can evert, and it's got two pincer-like jaws. Can you see those pincer-like jaws coming out? Yeah. And then back behind it, you can see the Parapodia there. They're a great shot of those jaws. I remember as a kid digging up these. They were great for bait, but I used to like to sort of squeeze just behind the head and evert the pharynx to see the jaws. It was kind of cool. Maybe mean, but it was kind of cool. All right, so that's class Polychaeta, the Polychaetes. Now we're going to look at class Oligochaeta, the Oligochaetes. So poly means many. If you're in lab this morning, you can't answer this question. Mono means one, di means two, poly means many. What does oligo mean? Have a guess. Mono means one, di means two. Then we've got oli, poly means many. Sorry?
Maybe like a group.
A bunch, some. Do you remember you talked about oligosaccharides? Monosaccharides, disaccharides, oligosaccharides, polysaccharides. So oligochaetes then, they don't have many bristles per segment. They've got some bristles per segment. All right. So they don't have a lot of these setae. Okay, just some. And they also don't have parapodia, or at least not marked or well-defined parapodia. No parapodia. So, easy way to distinguish oligochaetes from polychaetes then. And the classic example of an oligochaete is an earthworm. Now, those of you in lab this morning, did you feel the setae when you held it? Did you feel the roughness of the setae along the body? Yep, you can feel it. All right, so classic example is the earthworm then. And earthworms kind of eat their way through the soil. So they ingest soil, and soil's got a lot of organic matter in it, depending on the soil. The blacker it is, usually the more organic material. And earthworms can also come out of a burrow at night, and they can drag leaves down into the burrow. And the leaves start to decompose, and then they'll eat those leaves. But they'll sort of eat their way through the soil and organic matter. They break down and digest the organic matter in the soil and absorb the nutrients. Got a pretty well-formed gut. What's the name of the muscle contraction which moves food along? Remember the specific name of that rhythmic contraction of circular muscles to move a substance along a tube? Peristalsis. Good. Peristaltic contractions moves the food along. Lots of enzymes. Now they're very important in tilling the soil, it turns out. So they're important in pulling organic material into the soil. They're important in soil turnover. So organic farming practices encourage these earthworms. We want earthworms to be in the soil. Non-organic conventional forms of agriculture usually apply a lot of chemicals to kill primarily disease-causing worms in the soil, but as a side effect, they kill the earthworms. That's not good, and it degrades the health of the soil. So they're hermaphrodites, but they cross-fertilize. Now some can asexually reproduce. They simply break into two, and each part can develop into a separate individual. Some of them can do that. Now with earthworms, or at least with annelids, usually the closer to the head end you are, the more likely it is for the rear portion to develop a head. If you just break off those rear segments, there's less chance of that. Look at the size of that. That's a giant Australian earthworm. I'd love to know what kind of fish you'd catch with that. Huge, right? Isn't that awesome? If we could only get those to do our dissection, that'd be awesome. All right, so here's some diagrams then of what it's like inside an earthworm. You'll see it on the dissection that you do. Can you see the cursor on the screen? You can, good. All right, so obviously there's the mouth, there's the anus at the other end, and here's the gut, which goes the entire length. It's a tube within a tube, essentially. What's this cavity here? It's a coelom, coelomic cavity. Okay? So in red, we've got vessels. They have a very well-developed dorsal blood vessel, which runs the length of the gut, and a well-developed ventral blood vessel, which runs ventrally along the gut. And those two blood vessels are connected. Here it just shows main vessels, but there's lots of fine capillaries. And then at the front here, we've got 1, 2, 3, 4, 5 pairs of pseudo-hearts. Now don't think of these hearts as like your hearts, because they're not really. They are vessels that are thicker and muscular, and they squeeze and they help move the blood. And when you look at a live one, you can see a pulse of blood run along the animal in the dorsal vessel. Did you guys see it in lab this morning? Yeah, it was pretty obvious, wasn't it? The blood pulsing back in that dorsal vessel. All right. So what you can't see here are these, there are a bunch of yellow cells that surround the gut, which kind of have a role a bit like your liver. All right, we'll talk a bit more about those later. And in this diagram, we've got this in yellow, but in fact it's sort of a white, light tan colour, is the nervous tissue. So there are these cerebral ganglia, which looks like little bulbs like this around the pharynx. Did anybody see them? I know in the one I dissected, I saw them. You've got to go right to the front, anterior end, right around the pharynx. No? You've got to move some of the pharynx is sort of quite hairy on the outside. It looks hairy. You've got to move some of that to see these. But that encircles the digestive system, and then you've got these two nerve cords which run ventrally with small nerves that break off at each segment. Okay? Now, you did see these. I didn't point them out in lab, though. You've got these metanephridia. All right? And you're going to see them in each segment, and these are structures which have what kind of role? What do nephridia do? What would be the analogous organ in your body? The platyhelminths had protonephridia. Annelids have got metanephridia. What do they do? Sorry? Not gas exchange. Cast your mind back, dig deep. Anytime you hear the word nephridia or nephridium, you're going to think about one thing. Well, two really. It's analogous to your kidney. It's not a kidney, but it has a similar role. Salt and water balance. All right? Salt and water balance. And every segment has some metanephridia. So here's a close-up of one segment, and I love this diagram, I really do, it's awesome. It's a cross-section. In lab, you should have looked at the cross-section of the earthworm, and you're going to see this cross-section through the gut. This little fold in the gut. Why is there a fold in the gut? What's the function?
Contraction.
More surface area.
Good. Increased surface area. Anytime you see folding, if you're unsure of what the folding's for, you're going to give one answer. Increased surface area. It's typically why there's folding. So it's called a typhlossole, and it increases the surface area of the gut. All right, there's our dorsal vessel running dorsally, ventral vessel running ventrally. It's one way that you can orient a cross-section is the typhlossole. Now you know which is the dorsal and which is the ventral vessel. But the other huge clue is that these nerve cords, all right, in a cross-section, they kind of look like this in a cross-section. And you're going to see those on the ventral side. So you can orient, you can get dorsal and ventral side of your cross-section by knowing where the typhlossole is or recognizing that and secting through the nerve cords because they're on the ventral surface. All right, so you see the little bristles hanging out here and here. There's the CT, and again, you should have seen that in the cross-section. And there's our metanephridium, which is regulating salt and water balance. And then there's a septum between each segment, which is a very thin sheet of tissue, but it's quite tough. When we do our dissection, we cut along the body wall up here, and we've got to cut through each one of those septa. All right, and they're a little bit tough. Okay, everybody okay then? Yep. If you're wondering where you're going to get labels to label a cross-section of an earthworm, they're almost all right just there. So this is what the cross-section of the earthworm looked like, just like this. And again, here you can see the typhlossole is really exaggerated. There's our dorsal blood vessel, ventral blood vessel, there's our nerve cord. And you've got various muscle layers. So on the outside, you do have a cuticle, a really thin cuticle, not as thick as the nematode. Then you've got an epidermal layer, and you've got several muscle layers. Okay? All right. Good? Helps once you've seen it in lab. So that's all I want to say about the oligochaetes, all I want to say about an earthworm. All right? So we're going to move on to the leeches. Your book places them in the same class, the oligochaetes. In lab, I said that they could be grouped out to a different class called class Hirudinea. I'll give you that in lab. Don't worry about writing it down now. So you've all heard of leeches. You know what they are. Most of them live in fresh water. Some of them live in damp terrestrial environments. And there are a few marine. And they're predators of other animals. Parasites that primarily suck blood. Now, leeches don't really have any CT on them. CT help you burrow and move through soil. These guys don't burrow and move through soil. Don't need them. You can run your finger along and they feel very smooth. No parapodia. And they're very, very stretchy and strong. Let me tell you, leeches. You can stretch them a good amount. Have you had a leech on you?
Yeah, you can't get it off.
It's like a rubber band, isn't it?
Yeah.
It's like a rubber band stuck to your skin with super glue. All right? That's what it's like. It's pretty awesome, isn't it? Where did you get your leech?
I was at a lake in Idaho.
Was it?
I was playing in the water and I tried to get it off me with a rock and I couldn't even cut into it. It was so strong.
They're really tough. They're very rubbery. There you go. There's a leech attached to somebody's end of someone's finger. Do you think that hurts?
You don't feel it.
Doesn't. And I'll tell with you some reasons why it doesn't. All right, there's a teeny tiny little leech trying to attach, I guess, but it's not going to get through your thumbnail. All right. Oh, look at this. So these are the classic little leeches you might get in many sort of hot, humid, jungly areas of many developing countries. Madagascar, apparently, there's some areas in Madagascar which have tons of leeches. And if you walk through that area, you're going to get coated in leeches. So here's the huge medicinal leech. Isn't that a monster? Yeah. And they're still used today. So I'll share with you how they're used in a moment. So leeches then, of course, first they've got to attach and then they've got to cut through your skin. Human skin is remarkably tough. Have you ever watched yourself getting stitches? Even the needle has a hard time going through. Human skin's very tough. And these are the blades, the mouth parts, made of chitin that the leech uses to sort of saw through your skin to get into your blood system so it can suck the blood. So this is a leech sock. It's meant to keep leeches out. But what I'm told is that it doesn't matter what you try, you'll get leeches. They'll find any little like breach, any little crack, and they'll get in there and they'll find your skin and they'll suck the blood.
Sir Anthony Hopkins. Every actor at some point considers selling out.
I would never tarnish my name by selling you something.
That was kind of fun. Bring back memories. That's pretty cool. Do you think that would hurt? Those big jaws, knife-like mouth parts, cutting into your skin, yeah?
Nope.
Can't feel it. Why can't you feel it?
It is a
Mosquitoes?
So, in their saliva, they secrete a whole bunch of different substances. Some of them stop the blood from clotting, and some of them act as an anesthetic or something to stop the pain, so you can't even feel it. So, we've got this hirudo. How do you say that second one? Medicinalis. All right, Hirudo medicinalis is the big medicinal leech. Still used today, and they slit the skin with their jaw-like mouth parts. They don't have jaws. All right, we don't really get true jaws until we get to chordates. And they secrete an anesthetic at the bite site, so you don't feel any pain. And they also secrete an anticoagulant, so the blood doesn't clot. Now, the anticoagulant is a peptide. So what kind of a molecule is a peptide? What is it a polymer of?
amino acids.
Good, amino acids. All right, so it's a peptide made from amino acids, and it prevents your blood from clotting, and it's called hirudin. And we've used hirudin. I doubt we extract it from leeches. I would imagine, I don't know this for sure, but I would imagine that if it's a protein, there's a gene for it. And if we can find the gene for it, we can put the gene into yeast or bacteria. They will express the gene, and they'll make the hirudin protein for us. But it's used medically to dissolve blood clots. It inhibits thrombin, and thrombin's a key enzyme in the clotting cascade. And we'll look at the clotting cascade a bit later in the semester when we look at the blood and circulatory system. So here's a three-dimensional rendition of that peptide hirudin, and you can see these alpha helices and these beta pleated sheets, which are part of that overall three-dimensional structure. So they still use leeches today in medicine sometimes. Anybody know what they use them for?
I know when people have like a clot in certain areas, my mom, she's a nurse, so she would like apply leeches
Yeah.
to patients.
Sometimes under the skin you can have clots which won't dissolve or they won't go away by themselves and complicate ones. So they'll still use leeches today. They'll put them on, and they just happen to be much better at doing it than what we can. Yep. Okay.